Hey there, science fans! Today we're diving into the exciting world of electrophoresis. Imagine separating tiny molecules based on size and charge. That's what electrophoresis does. Think of it as a race for molecules. An electric field moves particles through a gel. Smaller charged molecules zip through quickly. This creates a unique pattern like a fingerprint. Scientists can identify molecules and learn about their properties. Now that we know what electrophoresis is, let's dive a little deeper into the science behind it. The key players in electrophoresis are ions, atoms or molecules with an electrical charge. When we apply an electric field to a solution containing ions, something amazing happens. The positively charged ions, or cations, are attracted to the negative electrode, called the cathode. Meanwhile, the negatively charged ions, or anions, move towards the positive electrode, known as the anode. The speed at which these ions travel isn't the same. It depends on two main factors: their size and charge. Smaller ions can navigate through the gel matrix more easily, so they move faster. This difference in migration speed is what makes electrophoresis so powerful. To perform electrophoresis, scientists use a special setup called an electrophoresis apparatus. The basic components include the power supply. This provides the electrical current that drives the movement of ions. The electrophoresis chamber, a container with two compartments, each holding an electrode, anode and cathode, immersed in a buffer solution. The gel casting system helps create a solid yet porous matrix for our ions to race through. These essential components work together to create the perfect environment for separating molecules based on their size and charge. We've mentioned gel quite a bit, but what exactly is it and why is it important in electrophoresis? The gel acts as a sieve, a porous medium that allows molecules to pass through while separating them based on their size. The most common type of gel used in electrophoresis is agarose gel, derived from seaweed. Another type is polyacrylamide gel, which has smaller pores, making it suitable for separating proteins or smaller DNA fragments. Besides gels, other media like capillary electrophoresis use a thin capillary tube filled with a liquid polymer solution. The choice of medium depends on the specific application and the size range of molecules we want to separate. Lights, camera, action. Running an electrophoresis experiment. First, we mix agarose powder with a buffer solution and heat it until it dissolves. We pour this solution into a casting tray with a comb to create wells. We mix our samples with a loading buffer and pipette them into the wells. We place the gel tray into the electrophoresis chamber and fill it with buffer solution. Finally, we stain the gel with dye to visualize the molecules under UV light. Section 6. Seeing is believing, staining, and visualizing results. Staining involves using special dyes that bind specifically to the molecules we want to visualize. Different stains target different types of molecules. Once stained, the molecules appear as distinct bands on the gel, with their position indicating their size and the intensity of the band reflecting their abundance. These stained gels provide valuable information about the composition of our samples, allowing scientists to identify specific molecules, compare their sizes, and even quantify their amounts. Section 7. Electrophoresis in action, real-world applications. Electrophoresis might seem like a complex laboratory technique, but its applications are far-reaching and have revolutionized various fields, including electrophoresis is the cornerstone of DNA fingerprinting, used in forensic science to identify criminals and determine paternity. Researchers use electrophoresis to study the structure and function of proteins, diagnose diseases like cancer and HIV, and develop new drugs. Doctors use electrophoresis to diagnose various medical conditions by analyzing proteins in blood serum. Electrophoresis helps ensure food safety by detecting contaminants like bacteria and allergens. These are just a few examples of how electrophoresis is making a difference in our world. Section 8. Conclusion: the enduring impact of electrophoresis. As we've seen, electrophoresis is a versatile and powerful technique that allows scientists to separate and analyze molecules based on their size and charge. This ability has revolutionized various fields, from medicine and genetics to forensics and food science. The enduring impact of electrophoresis lies in its ability to provide valuable insights into the complex world of molecules. Whether it's unraveling the mysteries of DNA, identifying disease markers, or ensuring food safety, electrophoresis continues to shape our understanding of life and contribute to advancements in countless fields.